Thank you. Good morning, and it's wonderful to be back and to see old friends and to meet some new ones. We're going to talk about the mouse model that a lot of you are going to hear about in a lot of these talks. I'm going to review some of the data that's been published as well as some of our new data and talk about how well this model recapitulates the human Barth syndrome. So we know the clinical features include cardiomyopathy, skeletal myopathy, neutropenia, growth delay, and prenatal loss from severe cardiomyopathy. This is the original Barth paper, and we know that it's due to mutations in tafazzin, which has been documented and is on the website by Iris Gonzalez, and that the monolysocardiolipin to cardiolipin ratio is increased. This, of course, was the first paper in Nature that documented the mutation tafazzin. We know a lot more now about the different mutations and splice variants. The information continues to come out. The models of Barth syndrome include, and we'll hear lots of talks about this, yeast, the iPSC-derived cardiomyocytes from Bill's lab, cultured ventricular myocytes, both mainly neonatal ventricular myocytes, Drosophila, zebrafish, and then we're going to talk about mouse because it is felt, of course, to be the most relevant. Although all of these models, and this was from a recent review that we wrote, have features of Barth syndrome, or whatever you want to call mitochondrial dysfunction, that have contributed to our knowledge of the pathogenesis. The goals of the mouse model should be, right, to exhibit an overt cardiac phenotype, and then we can test therapies. That's the bottom line. That is the mission of this foundation. We also want to be able to recapitulate cellular and molecular abnormalities that will then lead us to an understanding of the mechanisms that underlie this disease. Right? But the primary mission of this foundation really is to figure out therapies. So we know that this mouse model, that there are, sorry, it's a little bit small, but there's early embryonic lethality, there's this mild cardiomyopathy that Adam has talked about that takes a while to develop, actually, when we induce the tafazzin knockdown. We have the molecular abnormalities. If you look under EM, we have mitochondrial abnormalities. And this is the mouse everybody has talked about. So the first important point about this mouse model is that the published literature actually includes several different regimens of tafazzin knockdown by the doxycycline. So as originally reported in Zazaguch's lab, they used 625 milligrams of doxycycline per kilogram of mouse chow, and that was starting at early embryonic stage. Barry Burns' group then reported shortly thereafter their results, very similar actually to Zaza's, and they used a lower dose of doxycycline. It's 200 milligrams of doxy per kilogram of mouse chow, and this was prior to fertilization, so a little bit earlier. You wouldn't think it makes any difference. Who knows? We actually used what Taconic Artemis used. We used it in drinking water, not in the mouse chow, and we used 2 milligrams per mil. And we had figured out, based on typical intakes of what mice eat and drink, that our dosing was somewhere between 3 and 10 times the dosing of the other labs. This, in fact, may be very key to the differences that we see in the cardiac phenotypes. So the initial data, as I said, this is Zaza's initial paper in JBC. This is Barry's in human gene therapy, and ours came out in Journal of American Heart Association. These all came out within 1 or 2 years of each other. This is the key figure from Zaza's original paper that basically shows a dilated cardiomyopathy. Here's mouse MRI, and what it shows, first of all, it took about 8 months before you could see this mild cardiomyopathy. And it's a dilated cardiomyopathy with thinning of the myocardium. Barry Burns' lab showed a very similar thing with the lower dose of doxycycline. Here's MRI again, and here is this dilated cardiomyopathy with reduced ejection fraction and thinning of the myocardium. We didn't see any of that because none of our mice survived. We had universal prenatal and early perinatal lethality, but what we did see was this non-compaction cardiomyopathy present both in mouse fetuses as well as in newborns that we managed to catch before they died. But none of them survived. So we have this almost a dichotomy. We have this severe phenotype versus a very mild phenotype. And so some of the stuff that we've embarked on in the last couple of years was we wanted to see what well, what are the kinetics of the development of this cardiomyopathy? How long does it take? How short does it take? Can we alter it? Can we associate it with changes in the cardiolipin profile? And so our initial experiments are the control arm. I'll show you these data of bezafibrate experiments. So you'll hear the data from Min Dong a little bit later, but we have some experiments to treat the tafazzin knockdown mice with bezafibrate to see if we can get any benefit out of that. In these set of experiments, this is postnatal induction of tafazzin knockdown to avoid that prenatal lethality. We use the 2 milligrams per mil in the drinking water of doxycycline. This was generally started at about 3-4 months of age, and then we continued it and then followed these mice. We echoed them serially, and then at the end, 9 months after induction, we sacrificed them and did some biochemical analysis. This is actually similar to a recent paper coming out of Ed Lesnevsky's lab from VCU. They wanted to use this really inducible knockdown model to look at what happens in adult mice. So they actually took mice, and I'm only going to discuss this briefly because it yet is a different set of results. They started doxycycline at 2 months of age, so adult, young adult. They used 625 milligrams per kilogram of chow, and then they studied the mice 2 months later, only 2 months of induction, and their goal was to see if ischemia-reperfusion injury could be worsened in these tafazzin knockdown mice if there are differences in cardiolipin. Interestingly, they found that it didn't affect ischemia-reperfusion injury, number one. Number two, the other interesting thing they found was that monolysin cardiolipin to cardiolipin ratio was not changed. So the paper was kind of strange that way. I don't think Ed is at this meeting today. Oh, there you are. Ah, excellent. But I'm wondering if, in fact, the tafazzin deficiency was 40% down at 2 months. I'm wondering if it was just simply inadequate. But that's yet another set of results in this mouse that's a little bit inconsistent with the others. Okay, so what did we find in our control arm? And this is, again, postnatal induction in adult mice. The first thing, just as a general thing, is that the tafazzin mice weighed less. They are smaller. This is 3 months after doxycycline induction. This is 5 to 6 months. We pulled the data because we didn't think it was that different at 5 and 6 months. And then at 9 months, the mice basically didn't gain any weight all that time. The wild type mice did, and the differences are significant. What we did was, because the cardiomyopathy is mild, in fact, in our hands, we didn't see a cardiomyopathy at baseline, we decided to do stress echocardiography. So the way that we do this is we do pharmacologic stress. The mice are sedated under general anesthesia. We get a recording with echo at baseline, and then we inject isoproterenol intraperitoneally. And then immediately, pretty much, heart rate and contractility go up, and then we follow these out for several minutes. We used to follow them out for 20 minutes, but those experiments took way too long, so we then cut it off at 10 minutes. So the baseline heart rate at 3 months post-doxycycline is generally in the high 400s. As you can see, it's not any different between wild-type and TAZ mice. And then at 3 minutes post isoproterenol injection, the heart rate markedly has increased. And there's a little bit of a difference, and out at 10 minutes, it's pretty stable, but this is not statistically significant. If you wait a little longer, this is 5 to 6 months now post-doxy, the TAZ mice begin to show a difference and a statistically significant blunted heart rate response, and then the contractile response is also blunted. The heart rate response is also blunted at 9 months. If we look at the contraction, the fractional shortening, at 3 months, there's really no statistical difference, although you can see it's probably trending that way. At 5 to 6 months, it begins to show a difference here at 10 months post-stress, that the tafazzin mice just simply have a blunted, on average, a blunted contractile response. But interestingly, at baseline, they contract better. And this is unlike what Zaza's lab and Barry's lab have shown in the past. And then similarly, 9 months post-doxy, we have an even greater difference between the wild type and the TAZ knockdown mice. They have a clearly blunted contractile response, but they also started at a higher shortening fraction, which to us is puzzling. These are just some pictures of our echo, and the thing that I wanted to point out here is that if you look at a wild-type mouse, this is a typical adult, this is a typical juvenile, and this is a typical neonate. You can look for ventricular hypertrophy and you can index it. What we do is we look at the posterior wall and divide it by the left ventricular cavity. So we can basically normalize it to the size of the heart. A lot of people might look at the septum, but to me, after some consideration, I decided this is not the septum. The septum's over here. It depends on sort of where you echo the mouse. I thought the posterior wall might be a little more consistent. If you look at these hearts, the ratio, if you eyeball it, the ratio of the posterior wall to the left ventricular cavity is actually very similar. And in fact, if you look at the data over time in a wild-type mouse, that index, if you want to call it, we call it the LVH index, that ratio is very, very constant. It's about 0.2, the posterior wall to the left ventricular cavity. So one of the things that struck us was that these mice looked hypertrophied. The tafazzin knockdown mice look hypertrophied. At 3 months post-doxycycline, we did not yet see a difference. At 5 to 6 months post-induction TAZ knockdown, we saw quite a big difference, and then it was very consistent at 9 months. And it kept getting worse. So the LV seemed to get worse, similar, I suppose, to a hypertrophic cardiomyopathy. This is just some fun pictures. This is a mouse heart at baseline. It is a little bit thick. There's a TAZ. This is a little bit thicker than that wild type. And then this is at isoparenteral stress. The main thing I wanted to show is that there are also, on the EKG, ST changes, dramatic ST changes. The ST segments are nice and normal and flat here, but they're markedly elevated here. They can also be depressed. So one of the other things that we tend to see is that we see both arrhythmias in tafazzin knockdown mice under stress echo, and also either ST elevations or depressions, which goes along actually with some of the clinical stuff we see in cardiology clinic. A lot of the kids with cardiomyopathies will have ST changes even at baseline, and then they may become more dramatic when you exercise them. So that's postnatal doxycycline, that's postnatal tafazzin knockdown, and because the cardiomyopathy is so mild, we thought, and this is work currently funded by the foundation, we were wondering if we could build a better mouse. Something that lies in between the universal prenatal lethality and this mild late onset adult cardiomyopathy. So we wanted to minimize, we wanted to minimize that prenatal lethality, but we wanted to increase the incidence of an early Barth cardiomyopathy and just make it more dramatic, make it more overt. So we decided to try different doses of doxycycline prenatally, 0.25, 0.5, or 1 milligram per mil, in that range that would be somewhere a little bit higher than what Barry and Zaza had been trying, but lower than what we were trying. And then at weaning, we would actually increase it and try and maximize the cardiomyopathy after birth. And then we would perform stress echo at various ages. At 1 mg per mil, this is just from my Excel spreadsheet, basically no tafazzin mice were born. So 1 mg per mil did not seem to be a good dose. Everybody died. 0.25 and 0.5 mg per mil prenatally, we in fact had pretty much Mendelian survival. The 0.5 dose gave a slightly lower number of the TAZ mice, and we saw some runts. It was quite obvious. We had about 8 runts out of 50 or so pups. And so we decided to go with that, and the rest of the data are induced with 0.5 mg per mil in the drinking water of the doxy through gestation. It's like a couple days after plug, and then through gestation and through the weaning period. And then after weaning, we increase the dose. So I'm going to start with the hypertrophy index first. We're calling this the sweet spot experiments. And you can see now that by 3 months, we already have a statistically significant difference in the LVH, in the left ventricular hypertrophy, in the TAZ knockdown mice. And it's quite striking at 5 months, at only 5 months. This is more so than when we induce them postnatally. When we look at the heart rate response, this is a little bit busy, but I didn't want to separate this into 2 slides. But basically, the 3-month mice are the solid shapes, and the 5-month mice are the open shapes. And basically, we find a difference as early as about 3 months. The heart rate response is blunted. And same with the contractile response. It is blunted as early as 5 months. But again, the contractile response in the TAS mice the contraction seems to be better at baseline before isoparental stress, which again is a little bit puzzling. When we looked at the cardiolipin kinetics, we're trying to figure this out. This actually now makes sense. This is months of tafazzin knockdown of doxycycline, basically. At 1 month, if you feed these mice doxycycline for 1 month, the cardiolipin mass spec, the spectrum, really, it looks normal. It looks like wild type. At 2 months, you can start seeing bumps in the monolysin cardiolipin, and by 3 months, really, it's completely reversed and very similar to Barth syndrome. And if we graph this out, if we quantitate this and graph this out, by about 3 months, 2.5 to 3 months, the monolysis cardiolipin to cardiolipin ratio plateaus out and is the abnormality that is the signature for Barth syndrome. So it actually takes 2 to 3 months of tafazzin knockdown for the cardiolipin profile to switch over in these mice. So it makes sense that if you knock down, if you start feeding them doxycycline, it would actually take a while for the cardiolipin to switch over in the mitochondria, and therefore probably to see the cardiac phenotype. The other question that would be important to ask, especially if we're looking at gene therapy, is, well, what if we restore tafazzin? What if we stop the doxycycline and presumably restore tafazzin and therefore cardiolipin? Are these hearts irreversibly damaged, in other words? If we look here, this is actually an old slide from a couple of years ago. We've induced these mice for greater than 6 months, and then we took them off the doxycycline for 3 to 4 months. And the bottom line is, here's the off kinetics, and the tafazzin knockdown mice have a restored heart rate response to stress echo, and they have a restored contractile response under stress echo. So it looks like these hearts functionally go back to normal. So in summary, if we look at these cardiac phenotypes, the cardiomyopathy, we see decreased contractile force under stress echo. We need the stress really to unveil it. Zaza has some data where he further stresses them with isoparental subcutaneously. The interesting and puzzling thing is that some of the published data show that, as you would expect at baseline, when you look at these hearts, they're 8 or 9 months old, these mice, the baseline contraction, the ejection fraction or the shortening fraction, seems to be lower than in a wild-type mouse. That is exactly what you would expect for a cardiomyopathy. I have no idea why in our hands the contraction is stronger at baseline, although weaker under stress. But again, the induction protocol is a little bit different, and our hypertrophy is also different. That's down here. We see hypertrophy, whereas the other labs have seen a thinned out, dilated cardiomyopathy. The other cardiac phenotypes that we see, ST segment changes, elevation or depression. I didn't quantitate the data here in these slides, but it's at least 50%. 50 to 70% of our TAS mice under stress echo will have ST segment elevations. It's a much, much lower percentage in the wild type. Arrhythmias can occur, but they're not the ventricular arrhythmias that you might expect in a Barth patient. They don't kill these mice. Gets back to Adam's point that they survive, they have a normal longevity. And so the strengths of the mouse model seem to be, right, we've heard the data over and over again. The cellular and molecular changes seem to be very consistent. The lipid changes, oxygen consumption, things like metabolics, they seem to be very consistent. Adam's data show a pretty good similarity, right, in the proteomics between the mouse model and human Barth syndrome heart. The weaknesses are that the clinical features seem inconsistent. I don't know if it is strictly due to the tafazzin knockdown regimen, if it's due to how we feed these mice with the doxycycline. The cardiomyopathy is not particularly severe. It certainly doesn't seem to be lethal. I don't know if this is necessarily a bad thing, because we do have some phenotypic features, and we can test therapies, and we can use these as endpoints. Does the model recapitulate cellular and molecular abnormalities that lead to an understanding of the biology of Barth syndrome? I think absolutely. I think the answer to that is yes. Is there a utility to the TAZ knockdown inducible model? Again, I think the answer is yes. You can induce this at different points. You can change your doxycycline dosing. You can look at different aspects of cardiolipin biology using this system. There is, of course, we will hear just a little bit later, the doppelganger, the evil twin, as you will, the other mouse, and we're all excited to hear about Doug Strathy's data and to see if how much that mouse will add to this one. Just an acknowledgement, I work closely with Michael and Mindong and Young, who is also here in the audience somewhere. It's been a terrific collaboration over the last 6 months. We've had a number of students who have been supported by some of these foundations, but mostly we've been supported by the Barth Syndrome Foundation, the NIH. Thank you very much.
Great, thank you, Colin. We have time for a few questions. Barry, go ahead.
Thanks, Todd. Colin, great job in keeping this going to try to figure out exactly what that sweet spot is. And I think there are probably two things at work here in play. You highlight maybe that there's a developmental aspect that isn't seen when you induce TAZ knockdown in postnatal animals. So that's an important part of the biology, probably. So I think it's helpful to have that, but obviously if there's not enough compensatory functional reserve to have the mice survive, then that has a limitation in looking at therapies because you'd have to initiate them in utero. So can you speculate on what that might be? Why is it that this is an important part of the biology? Maybe Michael knows the answer. No. He's getting up for another reason.
So just getting back to the point of the prenatal stuff that we had seen, on the 0.5 doxy dosing, we actually have some histology, which I didn't show at weaning, and we did not see the non-compaction. I wonder if the non-compaction, therefore, is entirely lethal to this mouse if we were able to induce that. But, you know, trying to tweak this dosing would take years if you tried every single possible combination. I think that there are two different processes. We have some limited microarray data. I'd actually love to see some of the proteomic, non-metabolic proteomic data from Adam's lab. But if you look at prenatal hearts versus postnatally knocked down hearts, the microarray, the gene expression differences are entirely different. The functional annotation is entirely different. And so there are probably two different processes at play during heart development versus postnatal and adult life.
I think the similarity, though, to frataxin knockdown, which we've been studying in the lab over the past year, is interesting because there's similar energy deficits. It's knocked in the exact same locus with the same strategy targeting RNA depletion. And in those animals, when you do postnatal knockdown of frataxin, the mice die, which is not, so there is an important, obvious importance of the developmental effects that you've observed.
Colin, really nice work. I mean, these are the kinds of studies we all need to see, and not many of us, you know, don't embark on because of how difficult they could be and time-consuming. So this is great work. So with regard to the elevated fractional shortening at baseline, have you looked at? So I assume this is endocardial fractional shortening, right?
Yes.
Have you looked at mid-wall shortening? Or you're familiar with this term?
Oh, so like strain via speckle tracking or something.
Well, so it's calculated. So we see the same hypertrophy that you see. And I'm not sure if Zaza has seen it before the dilation, but that's a classic thing that you'll see in heart failure, right? Hypertrophy and then thinning. And it's known I used to study just heart failure and hypertension, but with LV wall thickening, early on what you see is sort of an artifactual increase in fractional shortening due to the thickening of the LV wall during systole.
Yes. That
will compress the endocardium closer,
yeah.
And give you this elevated endocardial fractional shortening. So there's a calculation you can do if you it's called mid-wall shortening, but essentially looks at the shortening of the midpoint of the LV that corrects for the thickening of the wall during systole.
No, we haven't looked at that.
Yeah, so that
would be good to look at.
Often we would see in our models that you get an elevation, sort of a hyperfunction, but in essence it's not enhanced contractile function. It's just a thickening of the wall.
Okay.
Ed Lesnevsky from VCU. So I would only like to second the comments about the developmental import, because on our reading of the literature, especially your JAHA paper, that really struck us. We were following up on, at the time, what we thought was an intriguing alternative splice product of TAZ that we saw in aging hearts, and that's a major interest of our lab, is role of cardiolipin in ischemia, reperfusion injury, and aging, and the combination. And so we were looking purposely to knock down TAZ in the adult heart to avoid the developmental aspects. And that's why I think the defects we saw were fairly modest. But I would point out, even at that 2-month period, we had a decrease in total cardiolipin content, and we had impacts on ROS generation from the electron transport chain and all. So I think a key role in these models clearly is the absence of TAZ, which is, of course, gone at 1 day once you start dox feeding.
Yes.
During development.
Right, so the mRNA is down very quickly. The protein takes about 3 weeks, I think we had figured out some time ago. The cardiolipin profiles will switch at about 2 to 3 months. Those are some of the kinetics. Bill?
Yeah, I just had a few comments. One, I think the dox sensitivity of this model, I mean, it's really frustrating and perplexing, but I think it points to, there's, obviously 2 possibilities. One is there's a really sensitive low threshold of TAZ that's sufficient to support the mouse, and we're not really, we're kind of near that threshold with the knockdown, but maybe not completely there. So basically we needed the genetic knockout, which hopefully we'll hear about.
Yeah.
But the other option is also that dox itself has toxic effects, particularly on mitochondria, and I think that's an important variable. Another variable is sucrose. So I don't know if you're adding sucrose to the water but obviously sucrose, first of all, changes the amount of water that the mice drink. And secondly, it can induce a metabolic-like syndrome that may also affect the phenotype. So I think that's, we've seen that the amount of sucrose you add actually has an important effect on the phenotype. The second point I wanted to make is your wild-type fractional shortenings are really low, and that makes me worried that you're masking the effect by lowering the wild-type fractional shortening.
Yeah, no, we've seen that, and I know that I'm blanking on her name up at Mass General. She's done a lot of mouse echo. Her point is that if you have wild-type mice whose fractional shortening is anywhere lower than 40%, you got a problem. I don't know. Every mouse I've ever imaged, over 15 or 20 years has had this kind of fractional shortening, probably from the anesthesia. We were talking about that at breakfast. So there are all these variables in these experiments. The doxycycline effects on mitochondria, yes, absolutely, that's possible. And yes, we do add sucrose, and that has also crossed our minds. But it's also in the wild types. But as you said, maybe it's the TAZ mice are more sensitive to the effect of sucrose.
Yeah, and the last thing, I don't know if you measured the heart weight of these mice for the hypertrophy, because this measure of hypertrophy you're using is very dynamic. I mean, in the same mouse, with ISO or not, you would get a different hypertrophy measurement. That's not really what we mean by hypertrophy. So I think the gold standard there is really to measure the weight of the heart.
Fair enough. Excellent. Thank you.